Welcome to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm Peter Bannattini, and here I discuss all things brain imaging with scientists from around the world. Today, my guest is Dr. Russ Poldrack, who has been so influential to the fields of fMRI, cognitive neuroscience, and brain imaging in general for the past 30 plus years. Russ is the Albert Ray Lang Professor of Psychology at Stanford University and Director of the Center for Open and Reproducible Science. Over the years, he has helped elevate how we do fMRI by creating resources and standards for sharing data and code. He's also working to advance the precision with which we think about task design and data interpretation through his Cognitive Atlas project, which is a knowledge base for cognitive neuroscience. Russ received his Bachelors in Psychology from Baylor University in 1989 and his Ph.D. from the University of Illinois, Urbana, Champaign in 1995. After a postdoc at Stanford, he started in 1999 as an assistant professor at Harvard University and Mass General Hospital. In 2002, he moved to UCLA, and then in 2009, he became director of the Imaging Research Center at the University of Texas at Austin. Finally, in 2014, he was recruited to Stanford, where he has been ever since. In this discussion, we look into some of the paradigm shifts in fMRI best practices that Russ helped foster, as well as some of the big picture challenges that we face when using brain imaging, modeling, and precision task design to derive new insights into brain organization and mechanisms of computation. Here, Russ also weighs in on the prospects of fMRI for biomarker derivation, as well as the exciting potential for single subject deep imaging. I mentioned to Russ that this was one of the fastest hours I've experienced in quite some time, as it was a completely engrossing discussion. I hope you enjoy it. Okay, Russ, thanks for joining us today. I think that of all the people I've interviewed, practically, you don't really need an introduction. Your influence has been so profound over the past, 30 years. I guess we're about the same age. I noticed that looking at your CV and everything. But and your influence, you've been, you started in cognitive neuroscience, and then you've sort of moved into a form of meta science, sort of trying to make the field better, and you've been quite effective in that, sort of leading the way in open data, sharing code, opening up how we interact with our data, such that hopefully that the quality of the data and the quality of the science can be improved. So just to begin, so let's start at the beginning. We're going to cover all kinds of things. Your history, how did you start? And you can go back as far as you want, as far as, when you first became interested in neuroscience, and
Sure.
What piqued your interest?
Cool. Yeah. Well, first, thanks for the invitation. It's really fun to be here. Well, I'll start close to the beginning. When I went to college, I knew I kind of always wanted to be a scientist as a kid, but I didn't know enough to know how to do that. So I thought the way to be a scientist was to be a physician. So I went to college and I was going to be pre-med. And then I kind of realized that, there was a lot of stuff there that just wasn't of interest to me, particularly my first chemistry class. At the same time, I started getting really interested in philosophy, actually. And so I kind of picked psychology as a major because it kind of sat halfway between my scientific interest and my philosophical interest. But I minored in philosophy and ended up, sort of thinking a lot about those sort of things. And the place I went to college didn't actually have anybody who was a cognitive psychologist, or they had some people doing like animal neuroscience stuff. But I was lucky to have a kind of a mentor who had Bud Barker, who had come up as an animal learning person, but he convinced me to kind of like think about, cognitive science, cognitive psychology. And my entree was really through reading kind of the philosophy of mind. I think the book that sort of cemented my interest in the area was Daniel Dennett's Brainstorms, which is a bunch of, kind of individual essays about different aspects of philosophy of mind. But it really kind of, got me excited about that. And so I still didn't really kind of know what I was doing. I'd never had a cognitive psychology class, but I applied to cognitive psychology graduate programs and ended up somehow getting into a couple of them. And so I went to University of Illinois. And first, I was just going to be a straight up cognitive psychologist and spent a couple of years doing that. And then Neil Cohen moved to Illinois, who was well known for his work in memory and amnesia. And I kind of got excited about that and joined his lab. So my first entree into neuroscience was really through kind of neuropsychological studies of patients. Though my dissertation ended up being pure cognitive psychology with some computational modeling stuff. So I came to Stanford to do a postdoc, and I didn't come here to do imaging at all. I came here to study patients. I was interested in skill acquisition and the role of the basal ganglia. And when I got here, this was 1995, so fMRI was just taking off in earnest at that point. Um, and Stanford was one of the hotbeds of interest at that point. And I got here, and the access to patients wasn't as good as I had expected, and at the same time, there was so much excitement about fMRI, and particularly it sort of appealed to my geeky side. I've been programming since I was in probably junior high, and really enjoyed kind of the computational side of it, and so I got kind of sucked into doing fMRI around 1996. And ended up being the primary focus of my postdoc and really of my career since then. I ended up having several faculty jobs. I went to Mass General first and had a position there for 3 years inside the Martino Center, or what was then the NMR Center. One of the places where fMRI was invented, if you will, and that was super fun. But then ended up moving to UCLA in 2003, and had fun there. And then I had an opportunity to move to University of Texas to start a new imaging center in 2009. Did that, and then had an opportunity to come back to the West Coast in 2014, and I've been here at Stanford ever since.
Okay, it's interesting. You mentioned a couple of things that sort of resonate with me. I had also started, kind of thinking along the lines of being an MD, but then and once again, it was sort of like the chemistry classes and organic chemistry classes, and I'm thinking, I'm not sure if I really want to do this. I'm more interested in and also I was sort of interested in philosophy, but I took the physics route, and then right, but I was still interested in the brain. I think I took a neuropsychology class, but then went physics. But it's interesting how paths sort of converge on certain things over time.
Indeed.
Um, I mean, as far as your first fMRI work is concerned, I mean, it was the standard. You collect. Back in the '90s, obviously, people collected 10 subjects, 20 subjects was considered a lot, and 100 subjects was considered overkill.
Right.
But then over time, the worry is that there were a lot of studies that were not necessarily replicable or reproducible. At what point in time, and we'll get into your work a little bit later, but at what point in time did it hit you that something needs to be done about this, and sort of along the lines of what particularly needed to be done?
Yeah. I think that the biggest kick was probably when Ed Vol's famous voodoo correlations paper started circulating. Before its publication, I think people were sending it around 2008 or so, and it highlighted this one particular kind of circular analysis practice that I realized we had been doing. That paper focused on kind of like social neuroscience, but a lot of people were doing exactly the same thing, and I realized we had done it. We ended up going back, and Jeanette Mumford and I kind of dug in and looked at how big of an effect did that actually have on our data, on our results. But that really highlighted to me the need for us to think a lot more about what we're doing with our analyses, and how they might or might not sort of lead us astray. Then a couple of years after that, the whole sort of reproducibility crisis in psychology really kicked in, I guess 2010, 2011, when people started paying a lot of attention to the fact that there were findings that were pretty clearly non-replicable. People started thinking about issues like p-hacking, and statistical power also became a big issue. That really became clear in 2013 with the Kate Button and colleagues published this power failure paper,
Yeah.
which highlighted the fact that across across a number of different aspects of neuroscience, not just imaging, that there was, low power, and highlighted the fact that, low power can lead you to have a high degree of, like, of false positive results.
Yeah.
And so all those things kind of came together to make me be really concerned. And I'm somebody who, wants to do something. And so, I asked myself, what can we do to try to make it better? And I've always been kind of, looking back, I mean, I was doing open source software, I had I think my earliest open source thing that I released was this SPM ROI toolbox back around like 2002 on, SourceForge, MATLAB code. And so I've always, thought that sort of, openness was a good idea. I've always been a fan of open source software. And this was, right around the time that people were starting to think that data sharing was actually a good thing to do. People had tried to do data sharing back around 2000 with the fMRI data center. That sort of, I think got hung up because the field wasn't ready for it.
Yep.
Around 20, I think the big the Biswal Milam PNAS paper was around 2010, I think, which basically showed, hey, look, if a bunch of people come together and amass data, you can answer questions you couldn't have answered with any of your little small data sets. And so we started the first sort of effort on our part was the Open fMRI project, which I think we launched around 2012 or so.
Okay.
Which aimed to share quote unquote raw fMRI data, and we can argue about what raw means, but yeah, and so that was that was really, I think my entree was really the Vull paper kind of getting me kicked off, and then the replication crisis kind of really pushing it.
Yeah. Yeah, and it's an interesting thing how to, sort of along the lines of how do you actually, I mean, you could just write a bunch of editorial papers or position papers about this and try to change the field, but actually trying to, and also certainly there's other avenues too, like, with OHBM, we're trying to do, best practices and then the Cobitus paper and things like that. But actually making tools and fostering large databases certainly help this. And so, you have, I mean, I just checking out your website. I mean, you have several things now that are up and running that are pretty much thriving. Open Neuro, which is the new data sharing platform, which is actually kind of the go-to place for sharing data sets. And you have other things that we'll talk about, the Cognitive Atlas, and then certainly with Chris Gorgolowski, and I guess Tal Yoconi, you started NeuroVault and NeuroSynth, which I remember Chris Gorgolowski talking to me about NeuroVault, and then the idea that you save your sort of statistical maps, and then all papers published in NeuroImage or even other journals are then scraped for this data, which is really, I found extremely useful as well in many ways. And then of course you've helped form, and I think it's really important, the standard for data sharing, BIDS. You were part of that effort as well. Which is really important. But all of these, it's hard because you're building this structure. You want to make the best use of it, and also, like, for instance, even with BIDS, you have a data structure, and certainly people have their own way of doing things, and some people might say it's not intuitive or whatever, but you have to agree on something. And
Right.
But it's really hard also because the field, how you acquire data, the quality of the data, or even subtle aspects of the data, that's all continuously changing. So you're trying to set up this structure and also be able to make it adapt to these changes as well, like as things become a little bit different. But
Yeah. And I mean, I think the success of BIDS, largely, kind of rides on the fact that, Chris Gorgolewski was, really good at engaging the community. Because, I think that these things, a failure mode for these kind of things is if it's like, viewed as one lab's project, right? And rather than something that the community owns. And I think Chris did a great job of making sure that it from the earliest part of the project, that everybody had a say, that we listened to all the voices. We didn't always do everything all the voices asked for, right? Because we also had a set of principles like, the sort of 80-20 rule, right? We wanted to make the thing work well for most people most of the time, for most use cases. And that requires that sometimes it's not going to work for some people, and we just have to kind of make peace with that. So I think one was like really being community minded. And then two was like thinking, as you said, thinking upfront about extensibility, right? The first, when we had the first bids meeting back in 2015, we were focused really only on MRI, on basically fMRI, structural MRI, and, maybe talking about diffusion, but diffusion wasn't really a big part of the discussion then. We built in an extension mechanism that was sort of inspired by this Python extension proposal mechanism, so that because we knew that things are going to change, as you said, and we need to be able to change the standard. We need to be able to extend it as new people come into the community. So most of the extensions to date have been for new data types, right? So we've added scalp EEG, intracranial EEG, PET, fNIRS now. And I think that's been, even though most, like if you go to Open Neuro, which Open Neuro, really the reason we step back a second, the reason that the big motivation for starting bids was, we had run Open fMRI, and to get data into Open fMRI, we had kind of our own bespoke data organization scheme. People would send us their data and we'd have to reorganize it into the data scheme.
Oh,
And
God.
the only way we could tell if we had successfully done it was we go run our analysis pipeline on it. And if the pipeline crashed, that meant we didn't have the scheme right. So we knew that we needed to we couldn't afford to hire enough people to do this for every data set. We needed to give people a way to organize the data themselves so that they could click one button and upload the data, right?
Yeah.
And so that's why having an automated validator that could basically look at a data set before it's uploaded and say, is it a valid BIDS data set? That was like central to what we did up front. And so then, so basically any data set that passes the validator can be uploaded to Open Neuro. So that means now PET data, intracranial EEG data, fNIRS data, all these different types of data can be uploaded without us having to really do anything other than update the validator to say, hey, PET is okay now.
Yeah. Yeah. Okay. Okay. Yeah. Oh my gosh, that would be right. That would be a ton of work to further curate the data once it's sent to you. Yeah, and certainly you have to do that. One has to be sort of aware of other things, like for instance, like I can even imagine like multi-echo data is a slightly
Yep.
different format or some additional file system that you might have to incorporate or something like that. But still, yeah, no,
Yep.
that's
Yep. Yeah. And I mean, to its credit, the bids community, I think, has been really good about thinking through how to deal with this stuff in an extensible way. So, they came up with a scheme for encoding, the necessary metadata around multi-echo
yeah,
so that you can easily share those. Yeah. An even bigger challenge is, if you want to share data that had been processed, right? What we call derivative data. By processed, I mean beyond just reconstruction of the images or whatever the scanner does, and then conversion into Nifty or one of the formats that we handle. That's the Wild West, right? There's the number. Fortunately, we're lucky. I mean, one of the reasons I think data sharing works so well in neuroimaging is because you know all the data come off the scanner digitally, right? We're not writing things down for the most part. I mean, we write a few things down in notebooks, but the data are for the most part, all digital. And we amazingly, thanks to the work of Bob Cox and others 20 something years ago, we had this standard Nifty, right? Which all the software pretty much handles. So, we're so far ahead there's a lot of other fields of science where they haven't even agreed on data formats yet, or their data formats are proprietary. So you have to buy an expensive piece of software even just to read the data. So
yeah,
I think we had a lot of advantages up front thanks to earlier community work.
yeah, yeah, yeah. I remember that. I remember that meeting. At least they had one of the meetings at the NIH where Bob Cox and Friston and Jonathan Cohen were sort of debating as to what the best format was, and yeah, I think that was early 2000s. Yeah, and that certainly these sort of things set the groundwork. So as far as open neuro is concerned, I mean it's growing rapidly. I mean you have right MRI, PET, MEG, EEG, and others. Um, so and the purpose, so right, the intended purpose is mostly there's many, and one is to sort of be able to have the data there so other groups can go in and reanalyze the data in their own way, or sort of look at something more subtle, or maybe compare or test analyzing, test new software. How has that worked out? Have people been downloading and reanalyzing, and how have there been any surprises or
Right.
things like that?
I mean, I think, we we have, the metrics suggest that it's getting a lot of downloads. I can't remember what the numbers are, but certainly terabytes have been downloaded in the last few years. In terms of surprises, I mean, I think, so one thing I should say up front is like, we had a reproducibility goal, but we also had a data aggregation goal, because I realized, around like 2009, we became really interested in sort of decoding across lots of different tasks, right?
Yep.
Sort of related to our cognitive atlas interests.
Yeah.
But we didn't think that we had the ability to go in and collect data on a lot of people across a lot of different tasks. And so part of the goal of Open Neuro was to have people aggregate all their data so that we can pull it together and try to start, kind of using it to do larger scale sort of analyses. We're finally starting to do some of that stuff, and I can say a bit about
Yeah.
that. But also the reproducibility angle was we thought an important one. The one pleasant surprise is that there've been, I can't think of any kind of at least high profile cases where of kind of gotcha sort of analyses where somebody took somebody's data set and came along and said, hey, you did this wrong. We can't replicate your data, blah, blah, blah. I think that, or, cases of people sort of complaining about being scooped on analyses or something like that. I think it's mostly been, I would say the biggest use cases have been people who want to develop novel analysis methods who don't have the ability to collect data themselves or the expertise to collect data themselves. They can just easily come and collect all sorts of different data. Aggregation. So people, want to analyze across multiple data sets on a particular task. And there's obviously, there's certain tasks where we have more data than others. But, for example, for the stop signal task, people have done analyses aggregating a number of different data sets across different studies. I think it's been really important for training. So if you want to put on a course and show people real data and have them work with real data, you can just grab the data easily from Open Neuro and do that. I mean, I'll just mention kind of one sort of larger scale thing that we've done recently that starts to, I think, really point at the utility of like these really big data sets. So, we've been inspired by the work going on in large language models, which have shown that, you can take these big models like GPT-3 and pre-train them on a ton of text, right? And then take those models and sort of do additional training on smaller data, right? And sort of leverage the representations learned in pre-training to then kind of fine tune them. So what Armin Thomas in my group did, this is a paper he presented at NeurIPS last year. He took a not all of Open Neuro. He took like, a little over 1,000 subjects, I think about 11,000 experimental runs worth of data from Open Neuro across all sorts of different tasks, like basically not worrying about what the task was, and just pre-trained these use architectures of like these transformer architectures like you're using large language models, where you're just learning to predict the next time point.
Yeah.
That's all. You just give it a bunch of data and say, for each of these time series, just learn to predict the next time point. And
Yes.
what that does is it trains it to learn representations of what fMRI data look like overall, right?
Yes.
Then he takes the representations learned in this model and uses it to develop decoders to, for example, let's take the human connectome data. We want to decode at any particular point what task is a person engaged in of the 20 something different tasks we might be able to pull out of that data set.
Yes.
And what he shows is that you can build decoders that are much more effective with much less data. Like you can build a decoder based on 20 subjects that works really well when you use the pre-trained model, whereas you're much worse if you just try to develop a decoder from scratch using the same data. So it starts to highlight, once we have these very large data, and that was only using probably less than 5% of the data, the relevant data from Open Neuro that he could have used, just because that was all we had pre-processed at that point. One thing we're doing right now is actually trying to go through and run as much data as possible on Open Neuro through fMRI prep to pre-process it, so that if people want pre-processed data for all these data sets, we can just hand it to them.
Okay. So that's really cool. So then the goal of this then would be to be able to put in any data set. I mean, I'm sure you've done this, without the paradigm, and actually predict what was the task in some sense.
Exactly.
So, and have you tried? This is something that sort of we're kind of trying to do is look at resting state. I mean, it would be interesting to apply this to resting state to see, transient thought processes.
Right. I think that's a really cool idea. We have not tried that yet. We've been pretty focused on sort of task stuff.
Yeah, yeah, yeah. So I'll hold this thought for a while before we go into the cognitive ontologies, because it does seem like this is directly relevant to that. Because you know you want to have this space of tasks, and you want to have this space of data, and you're trying to come up with relationships between the two and sort of build a structure. And yeah, I mean, it's sort of like the whole goal of fMRI in some sense. I mean, this is pretty much it. And I think, right, these decoders, this
Yeah.
GPT-3
Yeah.
type sort of things is
Yeah.
kind of
And
the way
the
to do it.
challenge, I mean, for us, this is something we've wanted to do for a long time. The challenge is, how do you need enough. I mean, obviously, you can get a long way using kind of self-supervised learning, like I mentioned, but ultimately, you need data that are actually annotated with what task is this person doing at this time, and then what are the cognitive components of that task, right? That's what the ontologies are meant to give us, right? Um,
Right.
and the problem is we've not had those kind of like detailed annotated data sets, and there's no automated way to do that right now. Somebody who understands the task has to go in and say, hey, I think this particular working memory task involves requires visual object recognition, and also involves working memory updating, or whatever cognitive functions we think might be going on. But
Right.
yeah, the goal, I mean, one way to frame the goal is, for any, I would like to be able to take a new cognitive task that you describe to me, and you say, hey, I'm going to do this particular task, and it involves these particular psychological functions. One way to think about this is that, we'd like to, for any particular new cognitive task that we've never seen before, if we know the cognitive functions that it engaged, we should be able to predict the activation map
Yeah.
for that new task based on what we know about how those cognitive functions are mapped to the brain from other tasks. And so we had a paper in Neuroimage, just I guess it came out last year, from John Walters in my group and others, um, where we got a start at this, using a data set that was developed by Jørn Diedrichsen and Rich Ivory and Maeve King and colleagues. It's called a multi-domain task battery data set. And it's a data set where you have individuals. Each individual, I think, had about 5 hours of data collection doing a bunch of different tasks. We ended up having like 40 something different task contrast. So we went through and analyzed each of those task contrasts in detail to figure out what psychological functions do we think are required to do this particular task. And then basically developed encoding models where we basically say, for this task, you have these particular cognitive functions engaged. For this task, you have these cognitive functions engaged. And then for every, we didn't do it on voxels. We did it in regions of the brain, like 1,000 regions.
Yeah.
In this region, how does it respond on each of those particular tasks? And then we can basically fit a model in that area to say, how is it responding to each of those cognitive functions? Then for some new task, we know what cognitive functions it engages. We can just basically invert the model and predict what should the activation map look like for that particular task. And it turns out we can do reasonably well. We can do reasonably well, at least within subjects. We can do not as well, but decently predicting from one subject to another.
So that brings to mind the challenge of the it seems to me that and you've talked about this before. You're trying to draw inferences about the cognitive functions associated with the brain activation, but it seems that there's potentially this inverse problem both ways. I mean, it seems like there's potentially the tasks might not uniquely map to the activation, and the activation might not uniquely map to the task in some regard. And so, yeah, it'd be interesting how to. I mean, maybe in the context of fMRI, that's not a problem for most things. But I mean, I don't know what your thoughts on that.
Yeah.
Yeah.
I think it's a really hard problem. I think you're exactly right that, well, certainly, tasks and even, what we think of as cognitive functions almost certainly don't map one to one to brain areas, right? Um, And it's also the case that, at least, I think this varies more across tasks, but, you know, from person to person, the way that a particular task maps back to the brain may differ, because of their expertise, because of the strategy that they use to perform the task. Some tasks can be done in different ways. There's lots of reasons that could differ across people. I think there's a deeper challenge, which is that, and this was also part of the motivation for starting to think about cognitive ontologies, 13, 14 years ago. We as psychologists have labels for cognitive functions. We think that there's a thing that we do called working memory maintenance, or a thing called phonological assembly, or whatever it might be, right? But those are just kind of human verbal labels for computations that are being done in brain tissue, right? And not, so the question is like, what, ultimately, if we think that it's an open question as to whether, a particular brain area can be even characterized as doing a single computation, right? It might well be that they do different computations when they're talking to different other parts of the brain, and, in terms of dynamics and so on. But, let's say that it does make sense to characterize a particular brain area in terms of a computation. The language in which we describe those computations is almost certainly not going to be, human natural language, right? It's going to be a computational language. And I think this has become really clear as people have started to try to understand and characterize how functions take place in artificial neural networks.
Yeah. Yep.
So, there was this kind of well-known paper a couple of years ago from Blake Richards and colleagues, which laid out this vision for basically, using artificial neural networks as sort of models of the brain, right?
Yeah. Yeah.
And that paper said pretty explicitly that trying to explain in human language what a particular part of a neural network is doing is a futile effort, right? That instead you need to think about it in terms of these things like, neural network architectures and learning rules and objective functions. That's something that I've been struggling with a lot, because I think there's almost certainly something right about it, right? That, the complexity of what our brains are doing and what our minds are doing is almost certainly not captured well by the kind of gross verbal labels that psychologists have come up with over the last 50 years.
Yeah.
But the question is like, how do we, what's the next step, right? I'm not satisfied by saying, oh, if I take a deep neural network and have it learn a task, and then I see that, units or regions in that network are predictive of activity in different parts of the brain, as people have been doing for a number of years. I'm not satisfied just stopping there and saying, hey, look, the thing that part of the network does maps onto that thing that the brain network does, but we can't say anything about what it is. That doesn't satisfy me, but the question is, how do you get from there to something that satisfies you without sacrificing, kind of fidelity to what the system's actually doing?
So right. I mean, it seems like the problem there. I mean, there's two sides of the problem. One is sort of defining the task as the computation, or the task itself, in a way that is more mechanistic in some sense. But it also seems that, I mean, I think about fMRI data. Can fMRI data, or even EEG data, or any of the data that we collect, actually get at what I mean, I often say, I don't even know what a computation looks like in terms of fMRI. And fMRI gives you, it gives you magnitude, it gives you some dynamics, it gives you, location. But is it possible? I mean, and I always, when I talk to people about using fMRI to inform models of what's going on, how deeply can fMRI data inform these models? And it's when you just get blobs, or maybe some features of activation, can you actually get at maybe what's the relevant scale to actually understand the
Yep.
the important mechanisms?
Yeah, no, I think that's a great question. And obviously, we've made those of us who've been doing fMRI for a while have kind of made a bet that this, relatively kind of like meso scale, few millimeters scale can tell us something useful. And I think that, there's certainly been some success using fMRI to do that. And even at the higher scale, right? The identification of these large scale networks that have come primarily starting, from resting state fMRI. I think that's
Yeah.
an interesting new discovery about brain function and brain functional organization that we didn't know about before, right? The question is like, how useful is that for understanding in more detail the actual computations that are going on? And I think that there's, what we know about complex systems in general, right, is they have this kind of multi-scale organization. And I think that, I still hope that there is something to be learned from, even if what we're looking at the mesoscale is sort of echoes of the computation that can only be fully described at the micro scale, that those echoes can still be useful in the same way that, that astronomers can use echoes of, cosmic microwave background or whatever to try to learn things about, the early universe, even though, those are nowhere near the kinds of measurements you would like to have to know in detail what was going on, seconds after the Big Bang.
Yeah. Yeah. So it seems that, right. I mean, it seems that you have these three levels. I mean, you have you want to make very precise ontologies and, and models of what's going on. You also have you also could have very low level sort of neural network models that you test to see how that might emerge to show this activation. Then you have your data in the middle, and it
Yeah.
seems that
Yeah.
some iteration between all those two levels,
Yeah. I
you
think
might be
that's
able to
right.
converge.
Yeah, I mean, this is one of the reasons that I've become really excited about, the work going on in AI and machine learning in the last few years is because, in psychology, we've always developed theories that are very kind of micro scale, right? They're theories of a very specific cognitive function, and the kinds of predictions that they make are, well, you're going to be a little bit slower on this kind of trial than this kind of trial. Or you're going to make these kind of errors, things like that, right? Um, None of those theories could actually do anything, right? They couldn't go out and actually perform a task, right? We finally have AI models that can actually do tasks, that the kind of tasks that we would ask a human subject to do. So, they're kind of stimulus computable end to end sorts of models. And most of them aren't good models for how, humans do things, right? Because they weren't built to be models of how humans do things. They were built to be models to actually go solve a problem that's solve an engineering problem, right? Go drive a car without running people over, or go answer questions, on a chat bot or things like that. But I think that those kind of models, nonetheless, they're almost certain. Like, let's take take GPT-3, right? Um, you can view GPT-3 as a model of how people reason or answer questions. It's not a very good one, right? It doesn't reason or answer questions in the way that humans do in all cases. But nonetheless, you could treat it as a theory and then say, hey, let's figure out, like, what does it take to get a better theory that's modified not to be better at benchmarks, but to be better at actually behaving like a human, and figure out what are the things that would need to change in that sort of architecture to behave more like a human. Um, and the cool thing about those kind of theories is then they allow you to basically start mapping, and the way that people have done on a smaller scale, for example, with like reinforcement learning models, you say you find signals from the model, like prediction error signals, and you ask where in the brain, do we see similar signals? Now we can start thinking about across these entire kind of end-to-end behaving models, how do they map onto brain function? Can we start to use them to kind of functionally or computationally characterize particular brain systems in the way that people have started doing in the in the vision world with convolutional neural networks?
Yep. Yeah. Yeah, exactly. And I think, right, I'm seeing more and more of that. Whereas, I even have a colleague, Eric Wong, who's, he's a physicist, but he's sort of playing with this too. And he's sort of like thinking, okay, well, maybe there's convolutional neural network models, maybe applied to visual cortex or maybe cerebellum. And then there's other types of models like, shallow associative neural networks that might be more relevant in cortex or
Yep.
whatever. And so,
Yep.
and yeah, I mean, I think it's exciting because I think there's a lot of work to be done, and it's interesting to make sense of also, like, there's a lot of people starting to, Danny Bassett is starting to, she has been for a long time trying to model fMRI data in some sense, and Randy McIntosh is doing dynamic modeling, and it's, there's there's sort of islands that are starting to, I think, just at the very beginning of sort of converging on a mechanism for thinking more deeply about this brain activation data. So it's not just
Yeah, and I think actually that bringing up Danny and Randy's work is a great point, right? Because one of the big kind of chasms I think right now is that there are people on the like take people from the kind of like neural networks world, right? They're some of them have started working on more kind of like recurrent architectures with dynamics, but a lot of the work in that world has been just like a feed forward networks that don't really have dynamics, right? People like Danny and Randy have focused a lot on dynamics and not so much on computations, right? So the network neuroscience approach has been to like take a brain and treat it as a bunch of nodes, right? And you don't care what the nodes do, right? You just care how they are connected to one another, and you're using the same type of models you might use to analyze a power grid or an airport connection network, like whatever the network might be. And so I think figuring out how do you bridge the insights from kind of network science with the insights from computational neuroscience and AI is a really critical one. I think there are people doing really good work along these lines, like people like David Cicillo and Laura Driscoll, who've been working on these sorts of things. But I think there's a lot more work to be done to figure out like how do you bring together the computational side with the kind of network neuroscience side.
yeah, that's a really good point. And I think that's, yeah, I mean, emphasizing that point of enhancing that sort of analysis is I haven't heard that discussed that much, and I think it's worth bringing out. And one thing that popped in my head, more nuts and bolts sort of thing, is with fMRI. I mean, it seems like a lot of these models. One slightly missing element with fMRI is that it's hard to differentiate, inhibition from excitation. Because it seems like so much of the brain organization is dependent upon this inhibitory and excitatory signals. It would be nice to be able to do that, and then who knows? We might figure out a way to do that in some way, but right now
Right.
that element's missing, and one doesn't know whether it's a
Yep.
critical
No,
element.
I agree. That's a fundamental problem.
Yeah, yeah. So just quickly back to the open neuro, and before we go to a little bit more, we talked a little bit more about ontologies, but with open neuro, so one thing that I see, and I think it's a really cool thing, but also at the same time, I'm not really worried about it, but that when I go to OHBM, it seems that the culture of OHBM has changed, such that I would say at least maybe a third of the young researchers there are, they've never collected data on their own. They're just analyzing data, and I think that's so cool, because I think data should be reanalyzed for new perspectives, and the methods it's very important. But at the same time, I'm sort of maybe old school in the sense that I feel like there's something gained from actually going about coming up with an experiment and collecting the data and getting a real sense of the source of artifacts, and it really tempers and tunes your interpretation of the data. If there are good standards, maybe that's not totally critical, but I'm just kind of curious what your thoughts on that are.
Yeah. I mean, I completely agree that well, one, I mean, I think that let me first say, um, I think that it's great that people who were not in a position to collect data can go do science, without having collected data. Um, and so and so that opens up science to a lot of people who wouldn't have been able to do anything, who might have like really interesting computational ideas or theoretical ideas. Having said that, I completely agree that without having collected the data, without having gone through the experience of collecting data oneself, there's just a set of things that you don't learn about data. And I think that it certainly is the case that I think people who've never collected data approach data in a more kind of rose colored glasses way than people who have, right? Because you realize when you've collected data, you realize all the things that can go wrong, right? Especially if it's like you collect data and then you do QA right away because you want to make sure things are working, right? And the experience of having done QA really changes how you think about data.
Yeah,
And so like in my lab, like, I've certainly had postdocs, and I'm kind of okay with postdocs who show up who say, "Hey, I'm gonna just work on data," right? For the most part, except for, like I certainly have some grad students in my lab who are just focused on the engineering side, right? They just want to develop methods. I don't insist that they get involved in data collection and designing experiments, but anybody who wants to do kind of neuroscientific work in my lab, I really, as a grad student, expect them not just to take data and play with them, but to really get involved in, developing experiments and kind of seeing the entire process. Because I agree that, that's it's a critical thing. I mean, one challenge, right, is that there's just, I mean, compared to when you and I started, right, there's just so much more to know right now, right?
Yeah.
For a grad student,
Yeah.
it's really daunting for them to both understand how to, like, design an experiment, all the psychological stuff, and to understand all the neuroscience side, and to understand all the MR side, and to understand all the analysis side, right? It's just, it's like I feel for them, right? Just because there's so much there. And so you might imagine at some point we end up sort of like physics, right? Where you have the experimentalist and the theorist. And then the important thing becomes that they know how to talk to one another, so that, the theorists and like, from talking with physicists, I get the understanding that even in that field, there's kind of complicated relationships between theorists and experimentalists, right? It's not as smooth as one might think from looking at it, from the outside. But I think that, that we do have to worry and think about, how do we address this challenge of just the amount that one has to know, and the limited time that one has to develop expertise. And if you spend 5 years in graduate school, you can't become expert in all the stuff, right? You have to kind
Yeah.
of say, what am I going to focus on? And then, and obviously there's a lot of stuff that I have published on that I'm not expert on, and I've been lucky to have collaborators who can kind of,
Yeah.
tell me what I'm doing wrong so that we can do it right. And I think that's you're always going to have to think about that, right? When I started doing fMRI, I had no idea I had no idea about MR, right? But I had Gary Glover across the street who I can collaborate with, who knew, knew a lot about MR. Um, and so I think we do have to think about, as things get more complex, just think about that collaborative model.
Yeah.
Even if we don't go to the full kind of like, you know, distinct, calling somebody a theorist or experimentalist, just making sure that if you don't understand something well enough that's important for the work, that you have collaborators who do.
Yeah. Yeah, I completely agree. I mean, yeah, it really is substantially more that people have to know. And they all have to, everyone knows how to program, everyone knows how to, work in that environment of, data formats and things like that.
Yep.
And yeah, yeah, and also at the same time, it's interesting because I also think that there's something about generating an experiment that is a creative sort of element too, that sort of forces you to think. Like, for instance, when you look at the data, and maybe people can just do this also when they're analyzing data too, is that, there's things like with fMRI, at least with fMRI, looking at things like the latency and looking at, you know, um, the various transients in the signal or the post undershoot or whatever that might be meaningful, that sometimes people, when they just go for the analysis of standard GLM or whatever, they might miss. And
Yep.
there's all kinds of interesting things that can be taught with, even without collecting the data though too. So
Yep.
um
Yeah. Yeah.
yeah, so
I
it's
mean, this has always worried me about, training programs that are called like cognitive neuroscience programs, right? Because I think that there's something really powerful that's gained by training in a specific field. Like, training for me, it was training in cognitive psychology. For you, it was training in physics. For other people, it might be training in physiology, where you really learn how to ask questions about a very specific domain, right, and develop experiments. And I think that often the programs that are like cognitive neuroscience training programs give people like superficial experience in all the different parts, but without that deep experience, you're not really kind of learning what it's like to develop experiments in a sort of a thoughtful way. And it's a challenge because, again, people need to know lots of stuff. So the question is, how do you get them to get that sort of more domain specific expertise in the context of this larger problem?
exactly. And I think you're right. That's the nature of a good graduate program, a good mentor that teaches
Yep.
them how to, this sort of art of asking, of asking the questions. I mean, it really is all about that. I mean, you can
Yep.
actually just go and do stuff,
Yeah.
and generate data and even papers.
Yeah.
But there really is, you always find certain people are asking just the right question in just the right way. Uh, uh,
Yeah.
it's still
And
kind of an art.
I also have to say that I still, even though my lab does increasingly more, kind of non-experimental work, either infrastructure work, analysis work, I have more fun sitting down with trainees and thinking up a new experiment and kind of, thinking through all the ways that it could go wrong and figuring out how to fix those ways that it's one of the funnest parts of science in general. And so missing out on that, I think is also just missing out on the, kind of the fun of actually, doing science.
Yeah, yeah, exactly. So a little bit about your cognitive atlas. I mean, I started. I have to admit, I haven't looked at that much in the past, but I've looked at it recently, and yeah, I'm trying to get my head around. We talked a little bit about this, but in terms of the way it looks right now, I mean, the way it looks right now is when I think of ontologies, I think of sort of like ways that you can structure new terms that are more precise or more mechanistic in terms of what the cognitive actions actually are, as opposed to things like, working memory or attention
Right, right,
or
right.
things like that.
So I think there's a zeroth order goal, which is sort of one of the reasons that we started the project was that going to cognitive neuroscience conferences and seeing people saying, "Oh, I have a task that measures working memory," right? And then seeing people measuring it in so many different ways, and not it being clear that they're measuring the same thing. So the idea was like, if we're gonna understand, how cognitive functions map to brain functions, right? Well, you know, there are there are mental things in our head that psychologists study, right? We can't see them. We can only elicit them and measure them via cognitive tasks, right? And so whenever we have a person do a task in a scanner, we think it's, evoking some particular set of cognitive functions. That's a theory about how that task gets done. It may or may not be right. Um,
Yes.
but unless we write down that theory, unless we know exactly what the mappings are meant to be between, task X and cognitive function Y, um, we can't start to then say, well, is it really the case that working memory maps onto this particular brain region or network, right? Because if that's true, then we should be able to take any task that supposedly taps into working memory and it should, evoke activity in that task. So one of the very early goals was just to basically say, what on, what are we trying to do with cognitive neuroscience, right? When we give people tasks, what are the things we think are being evoked? Because you can sort of see people nodding to those in their papers when they write about, in back task evoking working memory or whatever. But in psychology, there's been this longstanding ideas about how it is that you kind of define psychological constructs and then validate those constructs. Ideas going back to people like Paul Meehl in the 1950s, and those ideas have not been brought to bear on how we think about cognitive neuroscience studies. So one way to conceptualize what we're trying to do is really formalize what it is that people think they're measuring so
Yep.
that then we can actually go in and try to break it, right? It might be right in some ways. It might be that, that this thing that we call working memory, is really 12 different things and we're mashing them all together. But without kind of knowing all the without formally saying like, these are all the tasks that we think measure working memory. Now let's go see how they relate to one another. Then we can't really validate those relationships between sort of, brain systems and cognitive functions.
So when I look at this, it's like, right, there's a list of the operations, there's a list of the tasks. So right now, it's is there, like, for instance, with different types of working memory, how in that website in the cognitive atlas, then so you have like a listing then of all the types of things that might invoke, like, working memory. It's very comprehensive, and but I but I guess what I'm, it would be nice to sort of be able to ultimately is to is to simplify everything in some sense, to say,
Yep.
well, this, maybe working memory is just sustained attention to internal processes or something, whatever. It'd be nice to be able to sort of once you get this, to sort of be able to collapse it across certain principles of what's going on, and.
Yeah. Yeah. And we've always thought that, neuroscience data are one way to think about doing that, right? So, for example, if there's, some people say they have a task that measures working memory. Other people say they have a task that measures sustained attention. And if we see that those tasks systematically evoke similar patterns of brain activity, right, then that tells us that maybe those two constructs are not really distinct, right? Similarly, if you say, well, we're measuring working memory, and I look across the tasks that measure working memory, and I see that they systematically differ in their patterns of activity depending on like what else is going on, right? For example, let's say that some of them are doing working memory for one type of material and some for another type of material, and I see systematically different patterns, then that might tell me that there might be some distinctions between those things we call working memory. Or if we can find some degree of commonality, then we can start to pull apart what's common, what's distinct. Those are the kind of things that we ultimately would like to be able to do. The real challenge is mapping. So we have all these tasks listed in the cognitive atlas, and ultimately it's not necessarily the task, it's really the kind of the contrast, right? Because if I give you a Stroop task, right, there's a lot of different comparisons I might do. I might ask about activation for congruent trials versus rest, or I might ask about incongruent versus congruent. We're really mapping down to the individual contrast on the task, but then we need mappings between those and the data sets. So that's what, like in the paper I mentioned that John Walters did, we sat down and went and took each task and mapped it on, or each contrast and mapped it onto an entity, a contrast in the cognitive atlas. And that let us then do that analysis. But that was hours and hours of us reading papers, understanding the task, and then coming up with our best theory about how that works. Somebody else might come up with a different theory, right?
I mean, it almost seems that, I mean, this might also, if you can put the data in the right way, like you said, look at the contrast, and this also might be something for GPT-3 to, to sort of parse through this and come up with unifying principles.
I think that there's a lot of potential for, I don't think we know yet how well those types of models could work in much more kind of, like domain specific sorts of ways. For example, like saying, what task is this? What contrasts are being measured in this particular, study, right? And, unfortunately, to do that well, we need access to a lot of text, and a lot of text still lives behind paywalls. I don't think abstracts of papers are going to be sufficient to do that. Getting access to to all of that will be challenging, but I think, and we've been involved in some projects that have started trying to do that, like this NeuroQuery project with Gael Veracqua and colleagues, that has tried to mine text, relate it to coordinates rather than to actual brain maps. But if you could relate text to actual maps, for example, using NeuroVault, that would provide a much more powerful way to, I think, do this.
Yeah. So there could be much more discussion about that. But I just want to shift gears slightly because I think it was on Twitter, and I think in the all the discussion about the BWAS paper. I think your name came up, and I think that's how we decided it would be great to have you on. But so maybe let's talk a little bit about just a little bit about that, and maybe kind of curious what your take on that. I mean, certainly there's been a lot of discussion about the BWAS paper, and on one hand, people are saying, oh my gosh, fMRI is not sensitive because it takes 2,000 subjects to get this effect. I tend to argue, and other people tend to argue that maybe there were issues in terms of the homogeneity of the population being compared, the type of thing that they were using was looking at resting state, or I actually think that individual differences in structure and in functional organization, even spatially, how you actually try to align all this data smears things a lot too, and might reduce the effect size. So what's your thought on that? Especially relevant to certainly ultimately coming up with biomarkers or clinical applications.
Yeah. So I found the paper compelling. It didn't really tell us anything that you couldn't know from like statistical theory, right? If you know the effect size. So I guess the thing we learned from it is like, what are the realistic effect sizes with these kind of data, with these kind of behavioral phenotypes, right? And I totally agree that the imaging measures and the phenotypes may not be optimal, right? Nonetheless, people have been trying to do exactly those kind of studies with much smaller samples for a long time, right? So what it does is it gives us kind of like it tells us if you want to use the kinds of phenotypes and the kinds of imaging measures that people have been using in the literature for a while, this is what you can expect. This is the best you can hope to do. And maybe, maybe that particular data set wasn't exactly the right one, but I think, it seems generalizable across other data sets. In some sense, it's no different than what people in the genetics literature have learned over the last 20 years, right, with genome-wide association studies, that I think it makes sense that you wouldn't necessarily expect connectivity between two particular brain regions in a healthy population to account for a lot of variability between people in some particular psychological function, right? It's probably going to be something much more global. And so I think that, what my takeaway is we certainly can do better, and, along the lines that you mentioned. But it's I think it's more a kind of a cautionary tale about just taking what we have and easily and, doing large scale analyses and, coming up with quote unquote biomarkers. And so it certainly throws some cold water on that, but I hope that and I think they've been clear that, this does not impugn all of fMRI, right? And they've been actually very excited about, that group has been very excited about doing, much denser imaging on much smaller samples, right? And I think has shown that you can do that and get interesting new neuroscientific insights into, functional organization of the brain. So I think it's mostly just a cautionary tale about, about understanding, what are the plausible effect sizes you can hope to get in a study, and then realizing what power you need in order to get those.
Yeah.
Right. So it's almost certainly going to be the case that, with the data we have now and the analyses we have now, that there's a lot of studies that, that people probably had in their head that this particular finding says you should not go do that study with 100 subjects. Right. Um,
Right.
and so I think that it will be interesting to see if we, can figure out, for example, address the alignment issues, address the behavioral reliability issues, address the imaging reliability issues, address the homogeneity issues. If all those issues can be addressed, how well can you do? One would hope you can do better. But it may just be that fMRI is also, in terms of individual differences and kind of accounting for the differences between people, for at least for kind of like in the healthy population. Obviously, there are going to be, sort of like edge cases, tails of the distribution, just like there are in genetics, right? What we know now from genetics is that, there are rare variants that can have huge effects, right? Common
Yeah.
variants across people very rarely have sort of large effects in genetics, and that's probably the case in imaging too. In part just because, just as with genetics, those things are going to get, they're probably going to get filtered out by evolution. So I think while it does throw water on stuff, it also opens up a whole new set of questions about how can we improve our ability to find those kind of relationships.
Yeah, no, definitely. It illustrated to me that, right, there's maybe more I mean, certainly looking at fMRI data, you can actually get a significant effect from a single study, single run. And so you have, fMRI is very sensitive, but it seems that, right, there's potentially more variability, uh, among people. But yeah, so certainly it does open up the nature of the problem a little bit more clearly in this regard.
Indeed. Yeah. And I think it's good that it just forces, Tagliarconi had a paper that was actually in the same issue as the Vull paper back in 2009 that highlighted exactly this problem of the low reliability of correlations with small samples, right? And so it's, again, something we've known for a long time, and I think that this paper simply highlights it in a splashy way.
Yeah, a lot of work to be done as far as that's concerned, and certainly, I think that there is a lot of potential, which maybe we can talk on another podcast, of looking at individual differences, and certainly looking at. I mean, I don't think in this one we'll talk in depth. Maybe we'll mention it. So you're also, one of the things you did that was novel and bold, and sort of as you do, is your My Connectome study. Um, and I love that idea, and I think there should be more studies like that. Um, and in which you scanned yourself twice a week for how long was it?
With some breaks, about a year and a half.
About a year and a half, and I think that right, that data could be. I mean, it's probably being mined in many different ways. Is there
Yeah.
anything? What were the? I mean, there are many things you found from this, but I mean, is there still new information coming from that data as far as that's concerned?
Um, I mean, I think that it's probably, it's been mined so much now. There's at this point at least like 30 something papers that have used the data for lots of different things. I think it's largely probably at this point being used more for validation sorts of stuff than it is for kind of new discovery. But I think there's probably still is stuff in there to be mined. Um, I mean, if it's interesting because people often want to know, like, what did you learn about yourself from doing the study? And I can't say that like the data told me much about myself at all. Um, but I think that there's a couple. So in the paper that Tim Lauman wrote and published in 2015, based on those data, I think there were a couple really interesting things that came out of that I think have been probably the most impactful findings. One was simply, how much data do you need on an individual to get highly reliable estimates of functional connectivity? And I think that the analyses of that data set showed a lot more than people thought, right? So that's why a lot of groups are now collecting 30 minutes or more of functional data per person rather than the 5 to 10 minutes that they used to collect.
Yes. Yeah.
Um, I think the really the most novel, interesting finding, and this is not something that I or my team would have discovered. It was really Tim Lauman and Steve Peterson's group who sort of found this, was the sort of functional variance, right? So the idea that we all have this kind of global organization. So let me for those of you who don't kind of know the story, right? So um, I had started this study, and Steve Peterson heard about it, and we had a phone call, and he's like, "Hey, we're really interested in taking your data and using them to ask whether these like individualized parcellation tools we have are reliable, right? They've never had enough data on one person to do that. They wanted to take my data and do it. So we ended up with about, I think in that analysis, it was about 12 hours worth of data, something like that. And they started by just like splitting it into two and saying, if you do these parcellation tools, do you get these kind of like connectivity based parcellations? Do you get consistent parcellations across the splits? And the answer was yes, they're actually quite reliable. And then the next question is, what do the parcellations look like? And so if you do those on like smaller data, you see basically like everybody has like the medial prefrontal cortex is kind of mapping into the default mode network.
Yeah.
Right. When they did it on my data, they saw that for the most part, my medial prefrontal cortex mapped onto the default mode data, but there were a couple of parcels in there that were more strongly connected with other networks, like the salience network, right? And the question was like, am I just a weirdo? Is something wrong with my brain, or is that is that something that we see in everybody? And so, Nico Dozenbach's group, along with Steve and others, has gone on to really, I think, demonstrate conclusively that starting with their Midnight Scan Club and then additional analyses of like HCP, that everybody has these functional variants, right? They're all in different places for each individual. And that's why when you average a bunch of people together, you see, oh, the entire medial prefrontal cortex maps into default mode network. But it turns out that each of us has these little islands of sort of, different connectivity. And they've gone on to show that they seem to have some behavioral relevance.
Wow.
And so that's like something that nobody had ever seen until they had this amount of data. And so to the degree that I'm really proud of like one interesting novel finding, I think that's probably the one. The other thing that I think is really cool is that this has kind of moved other groups to completely change how they do their imaging, right? So,
Yeah.
I think Nico's group is one, Randy Buckner's group at Harvard now, like, is doing like, pretty much exclusively doing this kind of like really dense characterization of individuals.
Yep.
My group, all of the new imaging studies that we do now, we're collecting many hours of data per person.
Yeah.
Whereas, we used to collect, 1 hour long session probably. So I think it's changed the way that people do imaging as well. And I think that's a cool thing.
Yeah. Yeah. No, I think that there's, yeah, I totally agree with that. And I think that there's benefit from, there's sensitivity to be gained from looking at one single subject very, very deeply. And
And
yeah,
your group has certainly done great work along those lines as well. Yeah.
yeah, we've and we're still right. We're it's funny because that data set keeps on coming up in which we scan, somebody doing a very simple task for, in this case, right. It was over 10 hours, but uh, still, I mean, we're still trying to make sense of that as well.
Yeah, yeah, yeah. And so
Uh,
that's been another great example, I think, of how like just the availability of this kind of data set can completely change how. I mean, that data set changes how we think about activation and what it means, right? When the whole brain can basically activate for a simple task, pretty much, or deactivate, right?
right.
What does that tell us?
Yeah, and it tells you right. There's a lot more in the transients. I mean, doing simple. I mean, one thing that study actually told me is that, just modeling it with a simple hemodynamic response function is, I mean, the hemodynamic response is probably consistent, but the neural response is very potentially different depending on the area, and you might miss all that if
Yep.
you just have oversimplified models.
Yep.
So, yeah, I think that there's a lot to be gained from doing individual subjects over and over again. Before people just try to do broadly across many subjects. Um, yeah, so maybe on that note, this was the fastest hour and 15 minutes that I think I've spent in a long time. Maybe just to conclude this particular this episode right here, as far as the future is concerned, the more I think about fMRI, I mean, certainly there's many problems, and also other imaging techniques. There's certainly challenges, and there's certainly limits. We haven't even talked about clinical applications, but how optimistic are you? And how do you think the field will look? Not only in terms of maybe let's focus it on how much it will elucidate how we understand the brain, maybe in the next 10 years or so, and how will we
Yeah.
get
No,
there?
yeah. I think that's exactly the right question to ask. And I don't. I mean, I think I have mixed feelings, right? I think the thing that gives me optimism is in part the degree to which our field has, at least in part, been pretty self-reflective and self-critical and tried to not just kind of stick our heads in the sand when problems come up, but really figure them out, and address them, with, for example, things like, HCP and ABCD, right, which have come together to try to, amass large data sets, and whatever problems those studies may have, they're great examples of people, putting in a ton of work to basically generate data for other people to analyze, right? Rather than kind of holding on to it themselves and requiring people to collaborate with them, and all that sort of stuff. So I think they're great examples of our field really deciding that, we need to have the data to ask the questions that we want to ask. So I'm optimistic about that. I'm optimistic about, sort of coming together with people using other more invasive methods in animal models and figuring out how do we bridge the gap so that we can map what you can learn from mechanistic studies, and I think that now with the availability of, optogenetic fMRI in rodents, you start to be, even though obviously there are challenges, there are a lot of things we're never going to be able to map well between humans and rodents, but you still start to be able to ask a set of questions that you might not have been able to ask before.
Yeah.
Um, I guess, I mean, the landscape on kind of newer fMRI methods more than I do. I guess I still remain pretty skeptical that, within the next 10 years, we're going to have a broadly accessible fMRI method that's going to like fundamentally change, fundamentally overtake bold fMRI that everybody's doing right now. I think it'd be great if we could to get at other sorts of things. And then I think that the other thing that gives me optimism is the theoretical side, right? Having access to particularly the new theories from AI and machine learning that we can use to get more insight from the data we have. I mean, it's certainly going to be fundamentally limited, and I still remain pretty skeptical on, about whether it's going to ultimately be useful, for example, for clinical biomarkers at the individual level. And I think that there's probably a lot of like misplaced optimism about that right now.
Huh. Okay.
But ultimately, I think the way that our field has responded to these challenges and the kind of the ongoing development of new tools and models makes me somewhat hopeful.
Okay. All right. All right. Well, on that note, on that hopeful note, and I agree with literally everything you said, and is including the biomarker, tempered enthusiasm. As far as that's concerned, I think we have a lot of work to do to really understand even what that means and how to actually ask the right questions about that. With that note, yeah, I'd like to thank you for coming on. This has been great, and hopefully we'll have you back again because there's so much more we could talk about. So
Yeah, no, it's been a great pleasure, and I'd love to come back anytime.
great. All right, thank you.
Neurosalience is brought to you by Organization for Human Brain Mapping. This week's episode is produced by Ömer Faruk Gülban and Jeff Manter.